Welcome to the Deep Dive. We take stacks of incredible sources, pull out the gold, and give it straight to you. Today, we're really diving deep into the human brain, looking at conditions like autism, epilepsy, schizophrenia, and also the just revolutionary science around stem cells and these things called brain organoids. Our mission, to give you a shortcut, basically, to being properly informed about the latest neuroscience right from the cutting edge. You're going to get some, I think, really surprising facts, insights that might just shift how you think about brain development and, the future of treating these really complex disorders.
Yeah, what's truly fascinating, I think, is how our understanding of the brain, which was, let's face it, a bit of a black box for so long, is just being transformed right now. We'll get into why some of the older ideas about these conditions were, well, frankly, just wrong, and how these new technologies are finally letting us, directly see and even tweak the underlying biology, which of course raises this huge question. How much can we really copy and understand the human brain outside the body?
That's a great framing question. So, okay, let's start with one condition that gets a lot of airtime, autism. To begin, maybe just clinically speaking, what is autism? Our sources are clear, it's complex, defined by behavior.
Yeah.
It's a spectrum, and crucially, no single biological marker right now.
Exactly. That behavioral definition is key, and it's actually pretty common for most psychiatric disorders. And, while many people with autism live completely independent, functional lives, there is also a profound form, a form that can mean lifelong intensive care. It's really not one single thing. Think of it like the idea of fever back in the 19th century. It's a symptom, right? With potentially lots of different biological roots.
Right, and the prevalence. There's so much talk about rising autism rates. What's the story there? It's now close to, 3%? Which is huge compared to when I was in med school. It felt like a rare disease then.
Yeah, the numbers have undeniably changed. We've definitely seen shifts in the diagnostic criteria over the years. That's part of it. And also something called diagnostic migration, where, conditions that used to be labeled something else now fall under the autism umbrella. But honestly, the full reason for such a big jump, it's still a bit of a puzzle. Research is ongoing.
Okay, and the history of understanding it. I remember learning about the refrigerator mother idea, this awful theory blaming emotionally cold parents. Seems incredible now.
Oh, completely.
But then twin studies in the late 70s pointed to genetics, right? And now, like the last 10, 15 years, hundreds of genes identified.
Yes, that shift was, well, it was profound. Moving from that kind of psychoanalytic view to a genetic one, absolutely fundamental. We now know specific gene mutations are involved, things related to synapses, how neurons talk to each other. Also, problems with ion channels controlling electrical signals. We call those channelopathies. And even how DNA is packed, chromatinopathies, these are involved. And finding these specific molecular issues, that gives us actual targets for therapies. It's a huge paradigm shift. We're moving from just managing symptoms to potentially fixing the root biological cause, something we couldn't really imagine just a decade ago.
That's incredible.
And some really elegant work shows that, changes happening elsewhere in the body can actually mess with brain development too. It's complex.
Okay, another area people get confused about, the male to female ratio. Is it really 4-to-1, more males? What's going on there?
Generally, yes, the ratio holds around 1-to-4, more males affected. Now, there's discussion about potential underdiagnosis in females, maybe masking where symptoms present differently. But there are also likely real biological differences, how male and female brains mature, how they respond to stresses, especially around birth. Females, for instance, often do better with premature birth, hit certain developmental milestones a bit faster. So biology plays a role too.
Right. And then there are all these theories floating around. Diet, microbiome, the fever effect, where symptoms supposedly improve briefly. What's the science say?
Yeah, a lot of those are largely anecdotal, like the fever thing. Maybe someone nonverbal speaks a bit during a fever. It could just be general nervous system activation, There's no strong causal evidence. And look, improving sleep, diet, gut health, that can absolutely improve quality of life for anyone. We all struggle socially after a bad night's sleep, right?
Sure.
But there's zero evidence these things cause autism or that fixing them reverses the core condition. The overwhelming scientific consensus points strongly towards genetics.
Okay, so let's say a family gets a diagnosis today for profound autism. Best case scenario, infinite resources. What does the pathway look like?
Well, today, about 20% maybe will get a specific genetic diagnosis. A known mutated gene is found. Could be inherited, could be de novo, meaning a new mutation.
Right.
But unfortunately, even with that genetic diagnosis, It doesn't always point to a specific treatment, not yet. The other 80% or so fall into what we call the idiopathic category, meaning we don't know the precise cause. The real hope, the future, is moving towards individualized treatments based on these underlying causes, genetic or otherwise.
That really highlights this core problem, doesn't it? The brain's inaccessibility. Why has progress felt slower in psychiatry compared to, say, cardiology or oncology?
It really boils down to two main things, I think. First, just defining these disorders biologically. It's incredibly hard. Unlike a heart attack with clear biomarkers, psychiatric conditions are complex, defined by behavior. And second, maybe even more critical, is that the brain itself, especially during development, it's just fundamentally inaccessible. You can't easily biopsy a living, developing brain. If you look across medicine, the fields that advance fastest often dealt with more accessible organs. Think cancer, blood samples, biopsies.
Yeah, makes sense.
Yeah.
And that inaccessibility leads to all these correlations people latch onto, right? Like the old flu schizophrenia link or lower autism in Amish communities linked to lifestyle or vaccines. It gets confusing.
It gets very confusing for the public. And it's so important to stress, correlation is not causation. Just for the record, there is absolutely no solid epidemiological evidence linking vaccines to autism, period.
Right.
With the Amish community, yes, the incidence appears lower, but it does exist. And their restricted gene pool might also play a role. You know, the same core genetic mutations we find linked to autism, they're found all over the world in all sorts of populations.
Okay, so the brain's been this black box. How do we finally start prying it open? This is where things get really exciting, I think. Let's talk stem cells. This field just exploded, didn't it? There were ethical issues with embryonic stem cells, but then came this just absolutely brilliant idea.
Yeah, stem cells. Two key things, right? They can become other cell types, and you can keep them going indefinitely in the lab. The ethical debate around embryonic stem cells came from using fertilized eggs. That was a major hurdle. But then, almost 20 years ago now, Shinya Yamanaka had this incredible insight. He figured out how to, turn back the clock. He found 4 specific genetic factors, the Yamanaka factors, that if you put them into, say, an adult skin cell, they could reprogram it back to a pluripotent state, almost identical to an embryonic stem cell.
Wow.
These are called induced pluripotent stem cells, iPSCs, and suddenly you can make patient-specific stem cells without embryos. It just changed everything.
So hang on. You can take a skin cell from literally anyone
Anywhere.
and turn it into this master cell that can then become, what, heart cells, liver cells, brain cells?
Pretty much any cell type in the body, yeah. It felt like alchemy. It really did. For me, personally, this was huge. I was training, seeing patients with profound autism, feeling that frustration. We couldn't study their live brain cells. Yamanaka's discovery suddenly offered a way. Non-invasive, take a skin cell and maybe rebuild some of their brain circuits in a dish.
Incredible. Before we go deeper to that, quick question. Umbilical cord stem cells. People pay a lot to bank those. Are they the same kind of magic bullet?
Good question. Those cells are stem cells, yes, but they're already more restricted, mostly useful for blood disorders, things like leukemia. They're definitely not a universal fix for everything, and it's so important to distinguish that legitimate banking from these, frankly, dangerous, unregulated stem cell injection clinics, often overseas, claiming to treat things like autism. There's no scientific basis. It's incredibly risky. People have been seriously harmed. We often don't even know what they're injecting.
Okay, crucial distinction. So back to your work. You started building nervous systems in dish organoids, then assemblids. How did that start? What was the first step after iPSCs?
Right. So once we could make iPSCs reliably, the next big challenge was, how do you tell them to become neurons? Specifically, the right kind of neurons. We basically borrowed tricks from developmental biology, adding specific molecules at specific times to kind of nudge them along the right path. It became clear quite quickly we could make neurons. That wasn't the issue. The real question was, could we actually see disease-specific problems in neurons made from patients?
And this is where Timothy syndrome comes in, a severe genetic condition, autism epilepsy, caused by one tiny change in a calcium channel gene. What did you find when you made neurons from those patients?
Exactly. We took skin cells from Timothy syndrome patients, made iPSCs, then made neurons. And what we saw was subtle but clear. Their calcium channels, these tiny pores controlling calcium flow, they stayed open just a fraction of a second too long, letting a little bit too much calcium into the cell. That was it. The first specific defect we saw in patient-derived neurons.
Wow.
Was incredibly exciting, but, still very simple, just single neurons, basically, scattered flat on a plastic dish.
So how did you go from that flat dish to 3D
Yeah.
to these organoids. What problem were you trying to solve?
Well, the human cortex takes, 27 weeks to develop in the womb? A long time. In a 2D dish, neurons they don't survive that long. They don't organize correctly. It's not natural. We figured they needed to be able to float, to self-organize in 3 dimensions, kind of like little balls or bowls of cells. And when we managed that, getting them into 3D, it just unleashed totally new properties. We could keep them going for months, even years, watching them develop, forming layers, mimicking aspects of real brain development.
And what's just mind-blowing is that these organoids sitting in a dish, they seem to have their own internal clock. You mentioned seeing a change in NMDA receptors, these important signaling proteins that normally happens around birth.
Yeah.
But it happens spontaneously in the organoids after 9 months in the dish.
Yeah. It shows this incredible intrinsic timer baked into the cells themselves. We still don't fully grasp the mechanism and how they're keeping time, but figuring that out, imagine, could we potentially use that clock, maybe rewind it to rejuvenate old neurons, or fast forward it to study diseases of aging like Parkinson's or Alzheimer's much faster? It fundamentally allows us to watch human brain development playing out right there in the lab.
Amazing. But, okay, the brain isn't just cells, it's connections. Circuits. That led you to assemblids, right? What was the thinking there?
Exactly. For things like autism or schizophrenia, it's probably not that the cells themselves are missing. Usually, it's more about how they wire up, how they communicate in circuits. So, assemblids were the next step. The idea was, can we make different brain regions separately as organoids and then physically put them together, see if they'll connect? My lab's first one, around 2015, we took a cortical piece, mostly excitatory neurons, and fused it with a region called the medial ganglionic eminence, which makes inhibitory neurons. And we saw this beautiful stereotype migration of the inhibitory cells into the cortical piece, just like happens in development.
And you call them assemblids. Perfect name.
Yeah. My late colleague Ben Barr is a brilliant scientist. He really pushed me. He said, this isn't just an organoid anymore. You need a new name. So assemblid for assembled circuits. It seemed to fit. And it stuck.
So you can assemble regions. And what about function? How do you show these connections actually do something? That cortical spinal muscle assemblage sounds incredible.
Right. That was the key test. Could we build a circuit with a clear output? So we made cortical organoids. We made spinal cord organoids that contained motor neurons. And we made these little human muscle ball. 3 distinct parts. Now, the odds of a specific cortical neuron finding the right motor neuron, which then finds the muscle, all in a dish, you'd think it'd be incredibly low.
Yeah.
But they did it. Within weeks, we could put a little electrode on a cortical part, give it a zap.
And the muscle contracted.
And the muscle contracted. It showed the connections were specific. They were functional. From cortex to spinal cord to muscle.
That is just, wow. It really shows the power of self-organization, doesn't it? You make the building blocks, and they seem to know how to assemble themselves.
Exactly. They come with the instructions built in. It's quite profound when you see it. We've even built 4-part ones now, like a somatosensory assembly mimicking the touch pathway. Sensory neurons, spinal cord section, thalamus section, cortex section. And interestingly, the order you assemble them in matters. It reflects the developmental sequence.
So the therapeutic angle here. Can you use these to model and test treatments for things like chronic pain?
Absolutely. We've done that. We've modeled genetic pain disorders. There are mutations in sodium channels that can cause either horrific, constant pain or the complete inability to feel pain, which sounds okay, but is actually incredibly dangerous.
Right.
And using these more complex assemblids, we can see how the circuit-level interactions, often over long distances, are really key, and potentially how to intervene.
Okay, as these models get more complex, assembling brain regions, seeing function, even transplanting them, as we'll get to, ethical questions loom larger, don't they?
Yeah.
How do you navigate that?
Oh, constantly. It's something we think about a lot right from the start. Patient consent for the initial cells is paramount. Animal welfare in the transplant studies. And then there's the bigger question you alluded to, emergent properties. Could these circuits, as they get more complex, develop some form of learning?
Computation.
Either consciousness or sentience.
That's the big one.
It is, and it's why we're very careful with language. We deliberately avoid terms like mini-brains. It sounds catchy, but it's misleading, trivializes the science, and frankly scares people unnecessarily.
Wow.
We've actually led efforts in the field to establish clearer naming conventions, ethical guidelines, best practices to ensure we're communicating responsibly.
That's so important. Now, you mentioned transplantation. You've moved to putting these human organoids into live animal brains like rats. What does that in vivo environment add?
Well, As good as the dish models are, they're still missing things. Real blood flow, other cell types, complex signals you only get in a living system. And we found something striking. The neurons we grew in organoids in the dish, they were about 10 times smaller than neurons in an actual human brain.
Wow, that's a huge difference.
Huge. So we tried transplanting human cortical organoids into the brains of newborn rats. The timing is critical. Newborn brains are much more accommodating. And what happened was amazing. The human cells integrated. They got blood vessels growing into them from the rat brain, and they grew. They became 6 to 8 times larger than they were in the dish, much closer to their normal size.
So the environment is crucial.
Absolutely crucial, especially for seeing certain disease features. That neuron size problem in Timothy syndrome, we only saw it clearly after transplantation. And it's essential for testing drugs effectively. You need that more complete biological context.
Which leads to this incredible point. You're now nearing a clinical trial for Timothy syndrome.
Yes.
Based almost entirely on work done in these human stem cell models,
Right.
validated by the transplantation.
That's right. It's poised to be the first potential therapeutic for a psychiatric condition developed pretty much entirely using human stem cell models, then confirmed in vivo in our animal systems. We found a tiny piece of nucleic acid, basically, that can correct how that faulty calcium channel is processed. And it rescues all the defects we could measure. It's great.
Well, as my late colleague Lubert Stryer used to say, it feels like we're finally starting to demystify psychiatric disease bit by bit.
That's truly groundbreaking. What other conditions are you tackling with assemblids now?
We're working on some really tough forms of epilepsy, severe intellectual disability. Schizophrenia is a major focus, particularly a genetic form linked to the 22q11 deletion syndrome that carries about a 30% risk for schizophrenia, also autism, and also movement disorders like dystonia. Really devastating conditions. We're building loop assemblids to model the specific basal ganglia circuits thought to go wrong there.
And just to reiterate that important point you made earlier, your focus is on these profound, severe forms of autism, epilepsy, etc., not the broader spectrum where function might be very different. Precisely. That's a critical distinction. We're focused on the severe, often debilitating forms that need significant lifelong support. These are distinct from, say, higher functioning individuals on the autistic spectrum for whom it's often part of their identity. We might even need better words, eventually, to make that distinction clearer medically and socially.
This has been an incredible journey through this research. This deep dive really shows the arc, doesn't it? From these old speculative ideas about inaccessible organs to now pinpointing specific molecular problems and using this amazing self-organizing power of human stem cells, guided by really brilliant science, to build functional circuits in a dish. It really feels like this is where the future of understanding and treating the brain is heading.
It really does. And what strikes me is just the elegance of it, how these biological systems, if you give them the right starting parts and just some basic conditions, they know how to build themselves.
Yeah.
They recapitulate development in such remarkable ways. Connecting that to the bigger picture, it gives us this unprecedented window into the actual biology of these devastating brain disorders.
So here's a final thought, a question for you, our listener, to maybe ponder. Given this incredible progress, understanding the brain's own intrinsic clock, its power for self-organization, how might this deeper knowledge eventually empower us, not just to treat diseases, but maybe someday to fundamentally optimize our own cognitive health, our emotional well-being throughout our lives? Something to think about. Thank you so much for joining us on this deep dive. We really hope you're leaving feeling well-informed and maybe just a little bit awestruck by where neuroscience is going.